everybody. This is Terri Conneran with KRAS Kickers, and I see just a couple of participants. So we're going to hold on for just a minute as everybody starts coming in. So welcome. I'm Terri. I'm a 7-year lung cancer survivor and started the KRAS Kickers about 4 years ago. And I'm just kind of watching it kind of populate through. So until we actually get everybody in here, it just takes a hot minute for the magic of Zoom for us to kind of hang out and Zoom together. So I'm just really happy that you're able to join with us today. It's a beautiful Thursday. I'm in Charlotte, North Carolina, and I'm not gonna lie, ASCO was a real busy meeting. And it's about, I think it's 4 or 5 days. It feels like about a month of pounding the sidewalk and doing a lot of walking to hear a lot of the data that's going on. And I'm really going to be excited to talk with Dr. Lee today from Sloan Kettering and hear what his take is on all the excitement that's been going on in the KRAS space. But for right now, I'm just really delighted that you're here. And if I'm just rambling on, bear with me because I'm sitting here staring at myself. And so hang on, we're trying to get some things all worked up here with the sound check backstage. So if I could sing and dance, I would be doing that. However, I'm completely incapable. I have no skills, but I'm not afraid to be on screen and be stupid. So that's what you get with me. What can I say? You guys are here. Let me just tell you, I'm not sure if you thank you for the hearts. It's making me feel seen and heard. I'm going to tell you guys a little bit about how KRAS Kickers started. Real briefly, I'm going to keep it real brief. So 7 years ago, I was diagnosed with lung cancer. It was stage 3, and I went through treatment. It was chemo, surgery, kind of like standard of care. It was 2017, and then I had no disease, and started getting involved with lung cancer support groups and started hearing about these biomarker things. So I pushed my doctor, wanted to know what is my biomarker and what is driving my cancer. Like everybody else out there seemed to know what theirs was. And I felt like I wanted to belong too. I wanted to have a group of people and know what I needed to do. So so I was asking my doctor and I didn't know what my biomarker was. Super short story version is I had 2 recurrences. And even though I was asking the question, what was driving my cancer, I didn't find out till I went out for a second opinion that there was this KRAS thing. And it was on my report. It was on my initial report. And even though I was asking the questions, the doctor wasn't answering my question and didn't give me the information because it was unactionable. I'm personally a KRAS G12D patient. And subsequently, I've had a total of 5 different recurrences over the past total of 7 years. And I've been 3 years with no evidence of disease. Subsequently, I've been through all total. I have been through chemo surgery, which was a lower lobectomy. Oh, thank you. Lower lobectomy. I've been through radiation and an ablation. And I still get scanned. I'm here walking the walk. And okay, so since I have a screen, let me just tell you guys, the reason I even started this group is because there wasn't a group of people that I could connect with. You can't hear me? Okay, I'm told you guys can't hear me. Can you guys hear me? Somebody give me a thumbs up or something if you can hear me. I can hear you. Okay, cool. So you guys can hear me. All right, good. That's good to know. All right. So I forgot what I was saying. Oh, so the only reason I started KRAS Kickers is because I wanted to meet you guys. I wanted to know other people. I wanted to know other people that had KRAS, and I wanted to find out what it was I needed to know about KRAS. In fact, I had no idea what it was. how you said it or what it meant. And I think that that's really the most powerful thing that we've got here, is needing to know what KRAS is and what that means for our treatment. Subsequently, we've started doing and using KRAS as an acronym: knowledge plus research plus advocacy together equals survivorship. And that's all of our goals, is we need to be able to do this. And we're trying to get through and keep moving. Okay, I'm seeing it's Bob Lee. I see, oh, here he comes. Okay, all right, it looks like there he is. Hey, okay, sorry. I hope you weren't sitting over there listening to me rattling on, trying to fill out airspace. Thank you guys for listening to me, putting up and being here.
Hi, Terri. Hi, everyone. Great to see you.
Bob. Okay, so let me do all right. So hopefully, I don't know why my microphone's acting weird, and I see a storm coming, and I hope this is not a sign of things to come. So one more time, welcome. Thank you guys for hanging in there, listening to me rattling on and on. My name is Terri Conneran. I started KRAS Kickers. Today, I am absolutely delighted, and I'm not going to go through your whole long list of how incredibly important you are, Dr. Lee, but I assure you all that he just was one of the presenting speakers at ASCO, and he is like just so on top of what's going on strategically when it comes to the space. I need to make sure that I mention a thanks to the sponsors that allow us to not have to come chasing money from patients. Amgen, Genentech, AstraZeneca, Mirati, Veristem, Revolution Medicine, Sanofi, Novartis, and Loxo Lilly. Thank you guys for being able to be here. And now BMS, which used to be Mirati. So anyway, thank you, Dr. Lee, for being here today. And I'm sorry about the technical difficulties in the background.
Thank you so much, Terri. It's a great honor to be here. And thank you for your leadership at KRAS Kickers and the collaboration from all our members, not only in the United States, but all around the world. It is certainly an international movement that we're all a part of. I would like to kick off maybe with a brief presentation just to give everyone a flavor of what's going on in the American Society of Clinical Oncology ASCO annual meeting that just occurred in Chicago earlier this week. We did have a clinical science symposium specifically dedicated to kicking KRAS, and this was in the main stage auditorium, and several physician scientists, including myself, gave some oral presentations on the latest clinical trial results in this space, and maybe that will set the stage in terms of our discussions on what's to come and what we should be pushing towards in our fight against cancer. So I'll share with you just briefly a few oral presentation and abstracts from some pivotal clinical trials. The first one was presented by Professor Tony Mok at the Chinese University of Hong Kong, and Dr. Mok and a team of investigators looked at the phase 3 trial of adagrasib versus docetaxel in patients with KRAS G12C metastatic non-small cell lung cancer. This was conducted in a second line or beyond setting, so they previously had some standard chemotherapy and or immunotherapy. And this is an international trial that certainly is an important one in obtaining regulatory approval. Adagrasib, or KRAZATI, as many of you know, is an approved drug through the FDA accelerated approval mechanism for patients with KRAS G12C mutant lung cancers. And so this is a confirmatory phase 3 trial comparing adagrasib versus docetaxel, which is the standard chemotherapy in the second line or beyond setting for patients. So this is a big study looking at 453 patients. It's 2-to-1 randomization, which means it's a bit of a lottery when you get onto those phase 3 trials. 2 out of 3 chance you get adagrasib. and 1 out of 3 chance you get docetaxel chemotherapy. However, after completion of the either arm, you will still be able to switch to the other treatment. So everyone will get a chance to get all the treatment options. So if you were getting docetaxel first, and if you benefit, you continue. If there's no benefit, you then switch to adagrasib. So you'll get the best of both worlds. But this is a study to look at progression-free survival, how long this new drug can keep the cancer from growing. So this, as you can see, we've got a lot of patients randomized, 301 versus 152. And this is certainly an international effort. A lot of patients receive treatment. The patient characteristics are certainly fairly predictable. Patients in their 60s as the median, but we had young patients in their 34 or 45 on both arms, and as old as 80, 83. This was a study done with a heavy enrollment in Asia Pacific, but also it was an international study across multiple continents as well. And patients generally received prior chemotherapy or immunotherapy, whether together or sequentially. And as you can see also, most patients were former or current smokers, as consistent with the KRAS G12C lung cancer population. And this study allowed the presence of brain metastasis. As we know, this is very important for patients with lung cancer to develop drugs that will penetrate the brain metastasis. So this is the primary survival curve, so so-called Kaplan-Meier curve, looking at progression-free survival. So as time goes on, this is the percentage of patients who've still got their tumors under control. So you can see that there is a separation of the curves. The adagrasib arm did better than the docetaxel chemotherapy arm, although it's certainly a subject of debate in terms of whether this is significant enough. And so the median progression-free survival was 5.5 months for adagrasib and 3.8 months for docetaxel. I want to emphasize median is just one person, and we do have patients who actually take the drug for much longer than that. So this is just the middle person in that range of statistics. But overall, when you compare, there's a small benefit in terms of progression-free survival in terms of magnitude, but in terms of statistical significance, this was a hazard ratio of 5.58. So this is a relative reduction of 42% in terms of progression-free survival, in terms of progression. And the p-value 0.0001 just means that this is not by chance. This is a real difference. Whereas sometimes you can get a difference in numbers, but really it's like a fluke by chance. So that's why we put the p-value there as a statistical measure. So this is a real difference in drug efficacy in the pill of adagrasib versus docetaxel. And the overall survival benefit is certainly not mature yet, but you can see at 6 months, 45% of patients with adagrasib are still doing well versus 30% on docetaxel at 6 months. If you look at on subgroup analysis, it seems to benefit, favor adagrasib. You can see on this forest plot, we call it the forest plot because it looks like a tree, so it looks like a forest. And you can see the bars are towards the left-hand side, favoring adagrasib. If it's more towards the right-hand side, that's favoring the chemotherapy. So this is also another way of looking at the benefit of the pill versus the chemotherapy. And then you have the response rate. So how much shrinkage this drug produced in the tumor. As you can see, it's 32% versus 9%. Docetaxel, 9% is about historical, hasn't changed since the 1990s. It's about 10% response in chemotherapy in the second line, so which is not a very good statistic. So this is definitely an improvement. And the waterfall plot is basically the tumor shrinkage in the percentage. So if the bar comes down, it's like a waterfall, like a Niagara Falls. So that's a good result because you can see the tumor shrinkage in terms of percentage. If it all goes up, then that's like a fountain. So that's not good. That means tumors are growing. So fortunately, we're seeing a little bit more waterfall with adagrasib compared to docetaxel. The one at the bottom is sort of not looking so good, although there is certainly still benefiting chemotherapy. And intracranial response is also very important. This is looking at the ability to shrink the cancer in the brain. So if you have brain metastasis, it's very important that the drug gets in and kills the cancer in the brain. So you have a much better response, as you can see here. The waterfall on the top is adagrasib, whereas at the bottom, it's the docetaxel, which is questionable benefit. Still some benefit, better than nothing, but not looking as good. So overall, this study looked at also lung cancer symptoms. And in terms of symptom control, it's better with adagrasib compared to docetaxel as well. So safety, side effects, it's really on par. In fact, in some measures, it's looking better in terms of treatment discontinuation, 8% for the pill, 14% for chemotherapy in needing stopping the treatment, for instance. And then we have these are the potential side effects in terms of nausea, vomiting, diarrhea, elevation of enzymes. But overall, it's certainly on par with the chemotherapy in terms of the percentage of patients getting these potential side effects. But it is definitely better in terms of blood counts on the far right. With the pill, you don't get bone marrow suppression, anemia, and thrombocytopenia, which is low platelets and low blood counts, getting febrile neutropenia, infections, and hair loss. This is certainly better with the pill compared to the chemo. So in summary, this is a big study, and it showed that the adagrasib KRAS pill is better than docetaxel chemotherapy in terms of progression-free survival. That's the amount of time you control the cancer for. In terms of tumor shrinkage, it's better. Tumor shrinkage in the brain is better. Symptom control, it's better. And side effects were pretty similar. So this is the overall study outcome. And it's in many ways very similar to the sotorasib trial, which was published in The Lancet, and now it's a subject of great controversy whether it's truly better than chemotherapy. But it's certainly, in my opinion, as a physician who treats the patients, patients need to access as many options as possible. So having sotorasib as an option, having adagrasib as an option, these are all steps forward in the fight against cancer. And in terms of the magnitude of benefit, how much benefit is acceptable, well, we think further trials are necessary. So you can see that this is such an international effort with lots of countries involved, and that's really what it takes to bring a new innovation, a drug, a vaccine, any new therapeutics, any new breakthrough from the lab to the patient. This is what it takes. Otherwise, it takes 10, 15 years to develop one of those, which we don't have that. We don't have that many 10 to 15 years in our lifetime, and our patients need it today. So that's why it's very important to get together. There is a drug which is not actually available here in the United States, but it's developed in China and in collaboration with the European investigators. The fulzarasib is another drug, and this was done in combination with cetuximab. And it showed also, you see a waterfall plot, very early data, only 33 patients, but seeing already some substantial shrinkage across all patients. So this is more to come at an 81.8% response rate. So very impressive waterfall plot. This is cetuximab, is an EGFR antibody, plus this pill. So that's
for G12C also.
Yes, G12C. Yeah. So this is a G12C mutant lung cancer in the refractory setting, second line or beyond, but looking very promising, and definitely further development is needed. This is a combination trial with cetuximab, EGFR and KRAS together. This is a new drug from Lilly, olomorasib. They are selling it as the second generation KRAS G12C. What is second gen? What is next gen? This is a subject of debate in the oncology setting, but regardless, we are pleased to see a new inhibitor being developed for patients, and Eli Lilly is certainly very confident. That's why they're selling it as second gen. So we'll see. Let's see what data they are presenting. So this is from Professor Tim Burns from the University of Pittsburgh Medical Center, presented this phase 1b trial, specifically looking at olomorasib plus pembrolizumab as a combo. But you can see that they're also studying cetuximab. They're also studying pembrolizumab. They're studying many other combos with platinum chemotherapy. So there's more to come. But this particular presentation is looking at pembrolizumab as a PD-1 immune therapy combination with the KRAS G12C inhibitor, olomorasib. It's just got a name now. It used to be called LY with all the numbers. So as you know, when drugs get developed, they only first have a code. They don't have a name. And then once the phase 1 trial gets becoming more promising, they then will come up with a name. And then it's only when they get FDA approval, they have to come up with a brand for the market access. So that's how drugs get developed. So olomorasib plus pembrolizumab. So overall, you're looking at a small cohort of 48 plus 16 patients in the two cohorts with KRAS G12C non-small cell lung cancers. And these patients are treated both in the later line setting here and also in the frontline setting here. So two different cohorts. But over the 64 patients, you can see pretty similar patient characteristics, and some patients had prior treatment. Even some patients had prior G12C inhibitor with sotorasib or adagrasib. So this is the waterfall plot looking at first line. So this is treatment naive, de novo, upon diagnosis. Pretty impressive waterfall plot right here. And also the previously treated situation here. So you're seeing a pretty good response rate of 77% in the first line, 40% in the second line or beyond. So very early data, but it's showing that olomorasib and pembrolizumab is becoming feasible. Remember, with Amgen's sotorasib, the pembro combination had some problems. I myself presented that a couple of years ago at the World Conference on Lung Cancer and showed the liver toxicity issues with elevated AST, ALT, all these liver enzymes. And we tried all these mitigation strategies with dose reduction and lead-in. Now, these guys have learned it. So Eli Lilly learned from that experience. They have dose reduced. So this is given at a low dose at 50 milligrams or 100 milligrams twice a day. So it's not treated at the 100, 200 sort of dose. So it's at a low dose. And this is learning from the experience and trying to mitigate any liver toxicity. This is so-called a swimmer's plot. So it's like the Olympic Games. You have swimming in different lanes and, going to the right. And this is basically a plot showing how long this treatment can work for. So is it 2 months, 6 months, 12 months, 18 months? And the patients with the arrow on the right are basically showing they're still going. They're still going strong, and they're still taking the drug. So this, you can see here, the duration of response is fairly significant, and it's still going for most patients. So it's still early days. We don't know how long it'll last, but it's looking promising as we speak. In terms of side effects, it's not bad. In terms of grade 3 or more, you're seeing the elevation in liver enzymes being pretty in the low percentage, 8% here. 6%. Grade 3 is really the clinically significant ones you look for. Grade 1 and 2 often asymptomatic. Patients are doing well, really don't need to do much. So often it's just that enzymes, the numbers are a little bit high and it gets back to normal. But grade 3, you really have to keep an eye out for potential liver, significant liver toxicity. So, but those numbers are acceptable in the context of phase 1 trials. These numbers are actually quite good. So it just shows that at low doses, at 50 milligrams, 100 milligrams twice a day, you can actually get the drug in relatively safely without problems. And remember, phase 1 trials are mainly looking at safety first, but of course, we're interested in the efficacy. So these are sort of trends of AST and ALT, and not looking too bad, and patients are still going well per this swimmer's plot. So this then provides the basis for the phase 3 trial in the first-line setting upon diagnosis of metastatic KRAS G12C mutant non-small cell lung cancers. So this is looking at pembrolizumab plus olomorasib, so this regimen as the new first-line treatment option compared to the pembrolizumab plus placebo. But it's really, placebo is really nothing in it. So it's just pembrolizumab, which is the standard of care. And of course, they don't want you to have the placebo effect. So that's why they have to put the placebo in. So you don't know what you're actually taking. And docs don't know what you're taking. Only ultimately the study, the computer will tell you in the end what you're taking. But everyone will get the pembro, which is the proven standard of care. And then it's 1-to-1. 50% of patients will be on the olomorasib, and 50% will be on placebo plus pembrolizumab. So they'll all get some treatment, but it's just a matter of this combination or not. And then there's also for this is for PD-L1 high. For PD-L1 low, they're going to have chemotherapy plus olomorasib. So that's the current study, and they're looking at 384 patients here and 552 patients. So it's a big global study that Lilly is sponsoring, and certainly many sites, including MSK, are participating in this trial. So this will take a couple of years to read out. So we're looking at 2026, 2027. So we'll stay tuned. But this is the early data is looking fairly promising because of especially in the first line setting with good response rates and safety.
If I could just throw out there, Dr. Lee, just a reminder to folks that, excuse me, pembro is immunotherapy, and that's what they're combining it with, because it's still,
That's right.
It starts becoming a little bit of a blur.
Yeah. So as you know, pembro or Keytruda is a standard first-line treatment for patients with metastatic lung cancer. So if you have G12C mutation, they're trying to add this olomorasib onto the pembro to see if it improves outcomes compared to the current standard. So we're always trying to develop better things for patients, and this is the trial based on the evidence that was just presented from phase 1. So this is a phase 3 trial. And then finally, a presentation from yours truly on the sotorasib plus chemotherapy experience. As you know, sotorasib is the first-in-class KRAS G12C inhibitor, otherwise known as Lumakras. It's made by Amgen. It's the first one to get FDA approval as a KRAS inhibitor. And I presented the international phase 1 trial of this combined with chemotherapy. So here we looked at 37 patients in the first-line setting, which is just diagnosed, no prior treatment, and then 21 patients with second-line beyond. They would have had some prior treatment with chemotherapy or immunotherapy, and then getting on to this combination. So we did allow patients with brain metastasis that they just could not be clinically symptomatic. The demographics are very similar to the prior studies, median age in their 60s, current and former smokers, and we treated patients with in the first line with PD-L1 negative population. And this is the immune marker that predicts response to pembrolizumab or immunotherapy. But if you're PD-L1 negative, the immunotherapy doesn't work as well. So this presents an unmet need. And that's why combining chemotherapy may be an option for these patients. And in the second line beyond, we treated patients with a lot of patients with immunotherapy. 86% of patients received prior immunotherapy already. In terms of toxicity, this is very similar to chemotherapy side effects: anemia, low blood counts, low platelets, elevation of liver enzymes. In fact, if you treat in the first line, very few patients had elevation of liver enzymes compared to the later line. And I presumably because of the interaction with prior immunotherapy. Remember, immunotherapy and sotorasib combined may cause liver enzymes to go up. But if you treat in the first line, it's not really a problem. And in terms of the most common drug to be discontinued was actually pemetrexed chemotherapy. And so the sotorasib combo was deemed fairly safe. And in terms of treatment-related deaths, there was unfortunately one case, but that was treating a second line and due to febrile neutropenia, which is a very known toxicity from chemotherapy. So this was a chemotherapy-induced low blood count. You get infections. If you don't treat it on time, then this could be life-threatening. So this is one case of febrile neutropenia, but not known due to known chemotherapy side effects. So no surprises from this combination in terms of side effects. And then you have the sotorasib plus carbo-pem efficacy. We have 65% is a waterfall plot in the first line setting, and then 42% in the second line. So fairly similar to the numbers previously presented. Disease control rate of 100%. Majority of patients had tumor shrinkage, 94%. So very good responses. And in terms of durability, this is a swimmer's plot again, looking fairly good. We've got the duration of response median at 9.1 months. And most patients, actually 40% of patients are still swimming and going strong with the, you can see with these arrows. And so progression-free survival, this data after a median follow-up of 9 months, we finally got some progression-free survival data for the first time. And the median progression-free survival was 10.8 months in the first line. And then if you look at the PD-L1 negative population, remember these are the patients without the immune marker for response to immunotherapy, and therefore they have an unmet need. This regimen of sotorasib plus chemotherapy had a median progression-free survival of 11.9 months. So certainly looking pretty good for this population with unmet medical need. And traditionally, these patients don't do well. So it's looking even better than the general median. So certainly an option, a treatment option for these patients. And consequently, we're developing this combination in the phase 3. So that was phase 1 just presented. Phase 3 is already opening enrollment, including at Memorial Sloan Kettering, but global collaboration really needed. So we're having carboplatin, pemetrexed, and sotorasib, 4 cycles of chemo, and then followed by maintenance pemetrexed and sotorasib. This is in comparison to this current standard of care is just chemo and immuno combined and going on with maintenance chemo immuno. So this is the current standard of care. If you're not treated on the trial, this is the current approved, FDA approved regimen that's used worldwide. This is an attempt to do even better for patients, specifically looking at the PD-L1 negative population. So this is welcome because now we have a KRAS G12C inhibitor plus pembrolizumab, which is immunotherapy. You've got an immunotherapy combo in the PD-L1 positive situation. And then you've got the PD-L1 negative population. We have sotorasib plus chemo. So you've got the immuno combo, you've got the chemo combo. So these combos are being developed as new treatment options for patients in the first line. And you've got to hit the cancer hard in the first line. That's why this is a promising regimen for the future. And this also takes time. It would take another 2 years or so through international collaboration to get the results. Otherwise, these are 750 patients with over 37 countries. If it wasn't for that, it'll take us another 15 years. And we don't have 15 years. We need it now. So that's why it's extremely important to get the whole world together to do these kind of studies. And just a little plug for the international collaboration, working with multiple stakeholders at Bloomberg New Economy, with global regulators, pharmaceutical companies, patient advocacy, academic investigators and physicians, really trying to get global harmonization of clinical trials. If the standards can become uniform around the world, and every patient has a right to enter a clinical trial, then we're going to save. We did an analysis with McKinsey and Company, and that showed a 10 to 20% reduction. That is 1 to 2 million lives saved every single year if we can do this. Get the governments around the world to work together on fighting cancer rather than fighting each other. So this is a fight for humanity that we all belong to. So Cure for Cancer is an initiative and also a movement that we all are a part of, and really proud to have KRAS Kickers being a formal champion and member. And you can see Terri also participated and spoke at the plenary session of the Cure for Cancer inaugural conference in December, and really handed out all the KRAS Kickers socks and banners, and really spreading the movement around the world, not just in New York, but people from Latin America, from Africa, from Asia, really pushing forward. And a couple of Australian donors, Mr. and Mrs. Poach. And Mrs. Poach is a cancer fighter. She's unfortunately very sick with the metastatic gastrointestinal cancer, but really fighting, pushing hard, and her spirit shines bright around the world, and giving seed money and generous donations of $20 million to MSK, another $20 million to Sydney to really start international clinical trials and expanding access, not just in New York and Sydney, but throughout underserved communities. In Australia, we have Indigenous populations. We want to get clinical trials to them as well. And so they become access, they get access, and also they participate in the fight, and they will do their part to accelerate the global movement to cure cancer. And it's a public-private partnership recognized at the state level, Ambassador Rudd, Ambassador Kennedy, and of course, MSK pushing this forward. So, and we're bringing innovative technologies like liquid biopsy, which is a blood test that can detect KRAS on the blood test, instead of getting tissue pathology and infrastructure, which is difficult to do in Africa in many parts. And here we're bringing liquid biopsy to Africa as a leapfrog technology, so patients in the villages can also get a blood test and pick up KRAS and participate in a clinical trial. We can ship drugs to them, and they will contribute to accelerating that fight in developing new breakthroughs and new drugs and new diagnostics and prevention for patients. So we are partnering with Sophia Genetics and many other biopharmaceutical partners, working with the Nigerian government to bringing in MSK Access, a liquid biopsy test that can pick up many mutations, including KRAS, in Africa, in Nigeria, and by extension, the rest of the continent, eventually to leapfrog, and not as humanitarian work, but really trying to help them build capacity to be the engine for future of global oncology R&D. So MSK, where I work, is still New York focused. We've got some tri-state presence, and that's still the bulk of our operations. But we are also part of the global movement, and here we are partnering with cities and institutions around the world and trying to establish this hub and spoke infrastructure for international research development, empowering everyone to accelerate the cure for cancer. And with KRAS Kickers being a core member, really looking forward to the collaboration ahead to accelerate the cure for cancer. Thank you for listening.
Okay, thank you so much. That was fantastic. But boy, do we have questions. You're not done yet. So I hope you're going to stick around. Okay. All right. So first off, okay, because so much is happening around G12C, and we're going to move past G12C in 1 second. But I do want to ask specifically about the ramp 204, because it is Veristem, adagrasib, and I don't remember how to say the VS6766 for the ramp 204. And so I wanted to get your thoughts on that, because that is in the G12C space. And then from there, I want to kind of ask about a couple of the other things as well.
Great, thank you. So this belongs to the category of certainly combination therapy. We have the avutametinib, with Verastem Oncology, looking at combination with sotorasib and adagrasib, but certainly getting fast track designation, earlier in the year, and ramping this up with this vertical pathway blockade with RAF/MEK inhibitor with G12C. So can you shut off a few more checkpoints, a few more signaling pathways, or in the same signaling pathway, but certain more checkpoints, more nodes that you shut down? Can you shut down more completely? That's a very interesting hypothesis, and I think we just don't have the trial readout yet from that. I would encourage certainly clinical trial enrollment. However, this is not the only combination strategy. I mentioned EGFR as another combination, and we just showed that there's some promising activity, very early days, 30 patients, but still, we definitely need to expand that EGFR combination strategy as well. Whether it's cetuximab or panitumumab or others, I think that's a worthy pathway. SHP2 is another pathway that's really looking at the G12C off and on, that switch, and it basically shuts it off. And that's a promising strategy as well in the cancer biology of KRAS. So I think the SHP2 combination should be further developed. And there's also a new pathway. It's SOS1. So SOS1 is also on the other side of SHP2, and that's also another combination strategy. There are less companies making SOS1 inhibitors, but there are a couple. So I think there's certainly some trials happening in that space as well. So I think all of the combos are potentially worthwhile in developing, including the Verastem story. But to date, in terms of real data that's been published or presented, it's the chemo combo and the immuno combo so far for first line.
Okay, whoa, I can see major lightning going on out my window. So if I lose power, that's what's going on here. I mean, it almost looked like it hit me. Actually, it was very startling. Okay, so thank you for speaking to that. It might be a really good time to kind of like address why a G12C inhibitor won't work for somebody who's not G12C. Want to take a stab at that?
Yeah, of course. They're just structurally different. The groove of G12C, G12R, G12A, G12X, they're all slightly different. And so you have to, one way is to develop a molecule that fits into that groove and just locks it up. And that's certainly the G12C story, because when it goes into the off state, it basically opens up a pocket at the switch 2 region of the protein. Instead of a tennis ball, which is hard to lock with the key, it opens up a little groove, and then you put the drug in. It's a lock and key approach. It locks it up. So then it shuts down, and then you don't have signal and the cells die. So that's the lock and key approach. So D is a different pocket, R is a different pocket, and they're all different. And traditionally, they're very, very difficult to do lock and key because it's so round. But now we're discovering in various forms of shapes, they have grooves, they have these little openings, and then that's why we're developing these drugs. Now, D is a slightly different groove, and that's why we have a host of G12D inhibitors now being developed. And there's one through Mirati BMS. There's certainly one at Revolution Medicines, RMC 9805. That's a trial that I'm championing at Memorial Sloan Kettering. And there are certainly other mechanisms like protein degraders that's also looking at just shutting down the protein, not so on the surface of the protein rather than shutting down a groove. And also there's the pan-KRAS approach, which is so-called a molecular glue mechanism. So it relies on this chaperone protein including cyclophilin A, and basically it sort of, there's a protein that sort of binds to the KRAS protein and sort of makes it to turn on, but instead it glues onto it, onto the chaperone and onto the KRAS, so they're like stuck. And this molecular glue mechanism prevents the protein to become active. So that's a glue mechanism rather than lock and key. Lock and key, you got to be very pinpoint, very precise. Glue, you don't have to be that precise. So that's why it goes back to Terry's original question. G12C and D and X, can we just, inhibit it with the same drug? And then the answer is actually yes, because through the glue mechanism, it's not so much the lock and key anymore. You just glue it all together. And that is a way of shutting down KRAS, and the pan-KRAS or the pan-RAS inhibitors are being developed because of that mechanistic invention. Whereas the specific ones are lock and key. They're very much designed to the exact structure of the groove in the protein, and it has to be a perfect fit for the door to be locked.
Okay, so what I'm hearing you say, let's see if we've got this, okay, is the lock and key is if you're talking about something specific to, say, G12C or G12D. And
Yeah.
just because I live in the same, I don't know, style of house next to my neighbor, that doesn't mean my key is going to fit their house. Is that what we're talking about?
That's
so
exactly.
My G12D is different than a G12C, correct?
That's correct.
Okay, fantastic. And then the difference between a pan-RAS versus multi-RAS, take a stab.
Yeah, the pan-RAS and multi-RAS are very similar in the sense that you can develop the glue to just lock the door, and you just glue it together. So it doesn't, we're talking about locking the door rather than trying to open it, right? So you basically put a lot of glue, and even if the structure is a little bit different, you can sort of lock it all together, and glue it all together. And that's one approach in terms of a pan-RAS approach. Now, pan-KRAS is or multi-KRAS multi. If it's more specific to KRAS, that's a little bit more specific to KRAS, not NRAS or HRAS. These are the sort of junior partners. KRAS is still the king. It's the biggest. So when you have that specificity for KRAS, it's a difference in molecular configuration in terms of the glue. So it really glues up maybe part of that door, but there's some differences in the glue, in the exact location of the glue. It's a little bit more specific to the so-called switch to pocket. So it's more targeting that area. So I'm not sure what the best analogy is, but maybe you glue a certain point rather than just glue everything. So that's one way of targeting the KRAS.
Yeah, I'd rather glue the problem than glue my whole hand shut. So, right.
Right. Yeah. So there's a bit of debate, and people say, if you glue the whole thing together, you're going to glue your hand, and that may be more toxic. And maybe you just glue the KRAS and forget about NRAS and HRAS because they're junior players anyway. You go for the king, and maybe that's a better approach. These are a subject of academic debate. I think we need to do more trials in each approach, and there are many compounds in the pipeline for them.
Okay, so from a practical standpoint, okay, we talk molecularly, and at the end of the day, I really don't care how my drug works. I just want the drug to work, right? Okay.
Yeah.
That's just the truth. So
yeah.
I'm just going to start going through the Q&A. And so I'm going to kind of hit you kind of like random out of order because that's the way it came in. And so this is like a rapid fire. So let's rock and roll. Okay. So Oligio, okay. I can't say this word. G12V, oligometastatic. I don't know why I stumble over this word every single time, but I do. And you would never know English is my first language. So what would you say is coming? Is like a treatment plan? Because really, each one of us is really looking to make sure that we're strategically making good choices as far as what treatments we're on now and where we're going with it. So what would you say to that patient or that person?
Yeah, if it's G12V, I think the pan-RAS or the KRAS multi approaches are probably the most promising. In terms of the pan-RAS, there is the RMC 6236, which is now still the subject of phase 1 trials, and that is one that has produced promising early phase activity. Was presented at the European Society for Medical Oncology annual meeting in Madrid last year. And this is by my colleague Casey Arba at MSK. So this study is still ongoing, and this is certainly a promising approach. There are others, newer agents in the pipeline, and they still haven't entered clinical trial as far as I know, but many of them are in an effort of getting phase 1 activated. So, there's more to come in this space with both pan-RAS and KRAS multi. So I think this space is going to change in the next year or so, very, very quickly.
Do you think that's going to apply to things like G12R and kind of the lesser KRASs as well?
Yeah, absolutely. We've already seen promising activity in G12R and others with this approach.
Okay. All right. So we did answer that one. When looking for treatment for KRAS mutations, does that treatment have to be specific to the KRAS subtype?
Yeah, so you can do both. You can do the lock and key, which is very specific, right? It's only going to lock your door, not your neighbor's door. And so your neighbor's G12R, your G12D, you need a G12D inhibitor, not mix it together. So that's a lock and key approach, yes. But there is this glue approach. You just glue it all together. And that is a sort of a more KRAS multi, so it doesn't matter G12, G13, even Q61H, this is another variant of KRAS. They can all get locked up. So that's a more pan-KRAS approach that is now becoming quite promising. Now, which one's better? Well, I think this is anyone's guess. In my educated guess, the specific alleles are probably, it's so precision. I'm a believer of precision medicine, so the more precise, the better. But, I could be wrong. These things with clinical trial, you just have to enter it with a degree of humility. And sometimes it's best, maybe we should get the best of both worlds. Lock it up first. And then if the door opens because the cancer wants to bombard it, you glue it up again. So it's, have two shots against the KRAS rather than just relying on one. So, and what if we combine it one day? I don't know. And these are all very much interesting hypotheses that we have to test through clinical trials.
Well, and that kind of brings up a good point. How would I, as a patient, if you come to me, you're my doctor, and you're like, hey, here's like these 2 trials that you kind of qualify for. How am I, as a patient, going to make an informed decision to kind of like say, I would rather do this than that?
Yeah, I would participate firstly in a clinical trial that is readily available. I wouldn't wait, because there's so many unknowns with clinical trials, and I think it's actually more important to pick a trial that is going to be available. You're going to have a slot. It's going to be feasible in terms of transportation and your way of life. If it works, it works. Like you have to adjust your life, and it could work for a long time. You have to adjust your life accordingly. So I would try to participate in a trial as much as possible and still have the fallback option of standard of care. And so then you have the best of both worlds. In terms of picking trial A versus trial B, this is becoming a bit of a guess, and there's so many different factors. I don't think it's possible to have that crystal ball, and often, it's not relevant because the trial is not available or the slot is not available. So Yeah, I would probably focus on the practicals. You know, when is a slot available? Am I actually eligible? But if you go through all the screening and all that headache, and in the end you're not even eligible because of this particular contraindication or that. So I would try to get on a trial. Of course, you got to be molecularly targeted against G12, KRAS, G12C, G12D, or whatever your variant may be. It has to be a match. But if it's already a match, I think we have to enter it with a degree of humility and not say, okay, we've got 3 matches, I'm going to pick the best one. I don't think we have that crystal ball.
Well, and I gotta say, right? I mean, I didn't come from a medical background, and I was surprised, pleasantly surprised, that you have to qualify for a clinical trial. It's not like you guys are going back and coming up with some concoction. You're actually looking for a specific group of patients so you can have an intelligent scientific approach to this research. And so it really does make a difference. But as you're kind of looking at those trials, that's all the things that kind of factor in. What if you've already been on chemo, if you haven't been on chemo or immunotherapy or what have you, whatever your path has been. Is that pretty accurate?
Yeah, absolutely. And I can say that getting onto a trial is not easy, and we that's why the International Cancer Coalition work, you know, going beyond the academic silos, working with industry, working with government, working with patient advocacy, to change how we design clinical trial. It shouldn't be exclusive. It should be more inclusive of patients from all walks of life. So I think there's more work to be done to make the trial, you know, make it available to the patient. I want to bring it to the patient rather than having patients jump through hoops. The current reality is that it's hard to get into a clinical trial. Fewer than 5% of patients get onto a trial. 95% of patients miss out. So that's not acceptable when we consider clinical trials to be the best treatment to improve outcomes. So I think we certainly need to spread the word. Most people don't even know what a trial is, and most people are hesitant. You know, is this a lab rat experiment? And, you know, should I stick to the standard of care? Well, that's not scientifically the case. Scientifically, it's the best vetted treatment for the future. And the standard of care is treatment of the past. You need both. You need past and future. But I would go with the future if it's available. And this approach, you know, applies to the KRAS G12C, G12D, the KRAS multi. This approach applies to the on-switch RAS inhibitors, which actually is the another advance. And sometimes you can progress on an off-switch inhibitor and get onto an on-switch inhibitor. And there are several in development, and there are already trials being activated. So you can progress on an off-switch and go on to the on-switch. And that could maybe salvage the resistance and shrink the cancer. I have patients gone through multiple trials. So it's not so much mutually exclusive if you pick one trial over the next. In fact, you pick the trial that is a match for you at the time. And then if it doesn't work, you pick the next trial that's a match for you at the time. So they're not mutually exclusive, they're additive. And over time, yes, you buy some extra time, it's like 5 years on, I'm still alive, I'm doing well. And I just heard there are 5 different KRAS inhibitors that's come on board that wasn't available in 2019. You know, many of my patients will live through the first in human, you know, AMG 510 days when it just came on. You know, these patients are desperate. They're like progressed on chemotherapy, immunotherapy, there's nothing left. They've got hospice or some ineffective chemo. And then suddenly this AMG came on, you know, they come. And they're still alive today. They're still doing, they haven't, some of them progressed on AMG, but they're now more than just AMG. There's adagrasib, there's olomorasib, divarasib, on-switch inhibitors, KRAS multi. I'm going to have the patients go through all of them. And yes, some trials are more exclusionary than others, but, you know, there's got to be some trials that's going to be a match. So you go find the match. And it's not in exclusion to the next drug, because if that match gives you another 2 years, you take the 2 years. And come 2026, you're going to have more options by then. So then you're going to get more matches. So I see them as complementary and really additive, and we just keep going. And what about combination with we've already presented data on immune checkpoint inhibitors, but what about, all the vaccine stuff that's going on? And there's a lot of vaccine combos being developed right now, especially with mRNA. We're working with BioNTech on a lot of those combination stories. mRNA was initially developed for cancer, and then the virus came and became a thing of convenience. And it turned out that viruses are much easier than cancer. Yeah, we've got to go back to the main story of fighting cancer with mRNA. So I think these are all very promising, and I wouldn't just, exclude one trial over another. I would consider all of them. If it's a match, I give it a shot. What's the worst case scenario? I mean, it doesn't work for you, you come off it and you move on to the next one. And of course, you've got to find the right doctor, someone who you trust, and you build the collaboration, the communication. So if there's some serious toxicity, there's the early intervention plan to really salvage all of them. And as we saw with the toxicity data presented at ASCO, and yes, they're not benign, they're not just completely benign, there's some toxicity, but it's definitely not worse than chemotherapy. Chemotherapy has some serious toxicities as well. So therefore, in comparison, I think we need to give them all a chance. And that's why I have patients in practice who are living 5, 10, 15 years, even with metastatic disease. And these are generally patients who are very proactive in terms of getting on clinical trials, and generally people who continue to live as normal a life as possible. Just keep up doing what you love doing, whether it's traveling the world, whether it's looking after grandkids, whether it's writing, reading, or working. It's a lot of workaholics, they just continue to work. And yes, I say go for it. Don't take time off and work, and because they love their work and they feel really fulfilled, by doing that. So I have people who are really busy with work and still coming to the clinical trial, and they go back to work. So I have patients who are just incredibly, they enjoy traveling. So I mean, one patient I remember, booked the flight on the same day as my treatment, on the clinical trial, and he's worked it out that we had an 8-hour pharmacokinetics, and after that there's nothing that ties him down. So he's booked the flight on that evening, morning infusion, clinical trial, pharmacokinetics, all that monitoring, and then off he goes to the Greek islands, in the evening. So I mean, who am I to say you can't do that? So I think you just got to live your life, and nobody, and we all have a finite limit in terms of how long we get to live. I don't know, and nobody knows, right? Nobody knows, and therefore we just have to make the most out of the time we have, and just keep pushing. And I think clinical trial is very not only beneficial for the patient, but it's also very altruistic, because you're contributing to the greater fight for humanity. And if you win, you're going to bring a lot of hope for other patients. So I think it's a good thing just to just keep going with clinical trials.
Oh yeah, well, for sure. And I'm all for hope for other patients, but I kind of like it when I'm the one benefiting from it as well. It's okay. But I mean, I think that's part of it, is that we don't really realize how, it's not like, oh, I can start, like, say, I don't know, chemo or something, and then change gears as rapidly because of the qualification process, is kind of where I'm going with the clinical trials. And I think sometimes we miss that, especially when we're very scared up front with the treatment, and we just really want to have a plan. And so that's kind of one of the things I just want to make sure that we highlighted. And to that end, I also wanted to ask you about immunotherapy. I've seen kind of debates back and forth if you take immunotherapy before or after surgery. Do you have any thoughts on that for lung cancer?
Yeah. So I think this is a very exciting area in terms of perioperative treatment for lung cancer, colorectal cancer, many other cancers. Mm-hmm. I think increasingly combination therapy is the approach. And in lung cancer, combination chemo and immunotherapy is now becoming more and more a new standard of care. especially preoperatively, and then for some patients, we give it up post-operatively as well in terms of immunotherapy. Preoperatively, you have the benefit of seeing the response on the surgical resection specimen. And I'm certainly very encouraged that we are now seeing pathological complete responses in lung cancer surgery, which never really happened that much before. Whereas now a substantial proportion of lung cancer has been cut out, and you look at the microscope, they're all dead tissue, dead cells, no live cancers. It's all dead. It's still good to cut it out because you never know whether there's 1 or 2 cells hiding, but it's good. It's really encouraging to see under the microscope, they're all dead. So, and these patients do much better. And of course, we can then consolidate with further immunotherapy. And then the vaccine space is getting a little bit more interesting here as well. We combine the mRNA vaccine with chemotherapy, with immunotherapy in the adjuvant setting after surgery to prime the immune system to survey against any remnant cancers that might come back. This approach is now increasingly adopted in pancreatic cancer as well as colorectal cancer as well. So I think the mRNA vaccine trials at MSK is gaining ground, and we're working with BioNTech and others to look at these vaccine combinations for pancreatic cancer as well. So, and then what about the G12C and G12D inhibitors in that approach? Yes, we're also looking at not just chemoimmunotherapy, but looking at chemotherapy plus, say, sotorasib preoperatively before surgery. And given the exciting response rates that we're seeing with all the waterfall plots at this ASCO, can we use that not only in the metastatic setting, but preoperatively try to hit the cancers very hard, with this 70–80% shrinkage, and then we cut it all out? Can we increase cure in that regard? And that's definitely a very plausible hypothesis that may open up a new treatment option, a paradigm in the future. We're trying to do more about other alleles. G12D is still in phase 1 development, so we don't have as much clinical data, but we're already having, based on preclinical data in the mouse, and also borrowing the experience from G12C, we certainly want to combine that with EGFR inhibitors. As you know, for G12C, the addition of cetuximab or panitumumab is very important in colorectal cancer, G12C colorectal. Like the single agent alone is not enough for colorectal cancer. So you've got to combine it with an EGFR inhibitor, which is an antibody infused, once a week or once every 2 weeks, depending on the schedule. And that will markedly, more than double the response rate. So I think that approach may be adopted in G12D inhibitors. We just haven't got there yet. We have no data. We only have the mouse, but mouse and humans are different. So I think we need to do the trials and expand it, but we have good reasons to believe that combination therapy is necessary for colorectal cancers. And as you saw from the ASCO update, combination therapy is also needed in lung cancer. Even though single agent is approved for lung cancer, but moving into first line, combination therapies seem to be more effective. So I think that is the future. We're trying to do that the same for pancreatic cancer. G12D inhibitors, yes, we have this a specific G12D, several specific G12D inhibitors, and we're designing clinical trials as neoadjuvant, as maintenance, post-operative, and maybe one day all of those Whipples are going to be curative. I think our pancreatic surgeons would love to see that, because the vast majority of Whipples, you cut it out, they come back, and it's very sad. And Whipples is tough surgery. Patients have to endure a lot, and surgeons work very, very hard in these operations. It's like cutting out the monster, but then to only see it come back is disheartening. So perhaps we're at an era where we can give some combination G12D, which is a very important mutation for pancreatic cancer, to mop up and kill off these cells and then cut it all out so they don't come back. And that may be a cure for the majority of pancreatic cancers. I mean, that would be just a real breakthrough in the history of fighting cancer, because pancreatic cancer is arguably worse than lung cancer in terms of survival rates and all that. So I think, all of those advances, we need to learn from one another and not do it in silos. And that's why the approach in lung cancer is important for colorectal. There are some differences, but you can learn from one another. And the combination therapy may be different in colorectal and lung, but the general principles can be applied from one to another. And we're now putting pancreatic cancer on notice. So with this approach, we may be able to eradicate at least hopefully a substantial portion of pancreatic cancers. And then in terms of the primary and metastasis, I mean, that's a real biological question. In my experience, most of these tumors that are driven by KRAS, being so-called the king of oncogenes, they remain driven by KRAS. Even if they travel from the lung to the liver, it's not liver cancer, it's still lung cancer, but living in the liver, metastasized to the liver, and it's still driven by KRAS. So theoretically, if you shut down KRAS, they should all be killed off. And that is the oncogene theory. But I am seeing more and more heterogeneity. Sometimes they morph into something else. They have KRAS, but they have other genes that are driving the process. So it gets a bit more complicated. But generally speaking, KRAS remains the king of oncogenes, and it's still a very tough nut to crack. But we're really starting to make those cracks, and that's why I think KRAS Kickers is such a powerful symbol.
Thank you for that. I appreciate that. But it also, to your point, it's incredibly important for a patient to have their tumor retested after, when the therapy stops working, because there are new mutations that keep showing up. And there's co-mutations or, that may be showing as indication as far as like leading or suggesting we may benefit or have worse options.
Yeah, definitely there are more heterogeneous mutations coming up as the therapy evolves. And, I presented the KRAS resistance story, and we published the preliminary findings in Nature with Piro Lido and others a couple of years ago, and presented at ASCO, in fact, writing up a manuscript as we speak on that topic. And that's why combination therapy is needed to treat these alternate pathways. We talked about Veristem as one way to shut down the RAS-MEK-ERK pathway, but there are other pathways, mTOR pathway, EGFR pathways, and the SOS1 SHIP2 points in that oncogenesis. These are all very important to both shut down, but also to prevent the development of these pathways. And that's why the chemo combo or the IO, the immunotherapy combo are both good first-line strategies. And I think it's, these are going to be increasingly adopted worldwide. And that's why at ASCO, we just have, I forget, it was 30,000 or 40,000 people from around the world gathering, and just and really it takes the whole world getting together to push these trials forward. And I see some several attendees from everywhere. Tokyo, Japan, definitely needs to do more trials in Tokyo to push this forward. And we have great Japanese physician investigators leading the charge already. So looking forward to the to further breakthroughs from Japan and other parts of the world.
I agree with you more that worldwide, we should be able to come around to face the same enemy, which would be cancer, because nobody wants it anywhere. And so anything we can do will work along those lines. Okay, I'm going to hit you with a couple more questions. Let's see. There are a number of people that are on the RevMed, like the 6236 or one of the others. What would be just generally, okay, because obviously you can't give specifics, but generally, what would be like something that you'd be looking at next as a next line of therapy?
Yeah, so I think the 6236 RevMed, that's a KRAS, that's a pan-RAS inhibitor, and that's certainly a very promising one. It wouldn't preclude you from getting a allele-specific inhibitor after that. So that's like the general glue approach, but then you can do the lock and key approach after that. So you could try, if it's G12C, you can try sotorasib, adagrasib, or the trials of the omolorasib or the divarasib, and there's another one from Novartis as well. So there's quite a few of these that's already getting into the market, but also G12D specific inhibitors being developed as we speak, RMC 9805. So I don't think it's a problem to cycle from one another. There's also the on-switch inhibitors we alluded to earlier, that's so-called GTP inhibitors. So and that's there's quite a few. There's a RevMed one. There's the FMC as well, and that's also mentioned in the chat box. FMC, certainly they're looking at both on and off at the same time. So that's certainly an interesting approach, and I'd like to see data, especially when they already tried other KRAS inhibitors. So I'd rather cycle through multiple KRAS inhibitors if you can find a match, and that relies on eligibility, slots, the location, and all of those are not easy. I think if you find it, I'd go for it. Yeah, because then you have at least one option, a novel option that you didn't have before. And meanwhile, you still have the option of going on to others. So I would continue. I don't have anything specific for G13C, but the pan-KRAS approach and the pan-RAS approach or the KRAS multi would be a good fit. And oligometastatic disease, I would consider doing ablative radiotherapy to specific sites of metastasis, even surgery. I've taken out adrenal metastasis. I've had peritoneal metastasis taken out by surgeons. If it's truly oligo and you just continue the systemic medicine, whether it's KRAS inhibitor or others, you can continue that for quite some time. And I've had certainly successes through that approach with aggressive local therapy, and then optimizing the systemic medicine at the same time. So a lot of multidisciplinary collaboration is required.
Yeah, for sure. Okay, and then this is a good question around G12C. I'm taking a break and re-challenging with an inhibitor.
Yeah, I mean, I think you can get resensitization to the G12C inhibitor. So I would even cycle from sotorasib to adagrasib sometimes, even if one had already failed. But then you can maybe, after a period of time, you can try chemo or immunotherapy for a while, and then back into the KRAS inhibitor, because some of the clones of the KRAS cells may be more sensitized to inhibitor therapy already. So I think that's a viable approach in terms of switching. You have to sort of be a bit creative when you fight the enemy. You got to sort of hit it from a different angle sometimes. If you keep hitting one angle, it might get used to it. So you might want to go try something different, like a chemotherapy combo or an immunotherapy, a vaccine of some sort, cell therapy, TIL therapy, and then go back to the KRAS on or off switch inhibitor and hit it from this angle. And I've certainly anecdotally had lots of successes through that approach. So you can try multiple types of KRAS inhibitors and still get good response.
Certainly is a chess game, isn't it?
Yeah. And so I think we have to plan our moves, but also be very adaptive and not be so set in our ways. I think clinical trials in general open up a lot of opportunities, and I think the immunotherapy approach with cell therapy, TCRs, these are all very interesting, and we certainly can engineer molecules specific for these mutations, and that's some of them are opening in terms of early phase clinical trials. So we don't have any data yet, but preclinically they do seem very promising. So I think we're worth giving it a shot as well.
Oh, for sure. Okay, and I'm not familiar. A lot of times I know the like biology, but I don't know which KRAS variant ALA146THR is. I'm not sure which one that is offhand. Um, Somebody asked about are there any clinical trials specific to that? I don't know if you know that one off the top of your head or not. I'd have to look it up to see what those proteins are.
Well, I think this definitely does not fit into any of the allele specific um, uh,
Yeah.
KRAS inhibitors. However, you know, A146T mutation can still be, it's actually more common in colorectal cancers, but it certainly can be targeted through a KRAS multi approach. So I think that glue rather than the lock and key might be an approach. And that's still early days because the glue approach is still very early days. It's phase 1, so I don't think we've gotten into combination yet. And as you heard earlier, colorectal cancers generally require combination therapy. So, you know, I would certainly inquire about clinical trials, including the RMC 6236 and other new KRAS multi trials that are in the pipeline.
Yeah, a lot of those well, for the most part, clinical trials are, I don't want to say they're the newest. They're not old school thinking, and so there's such a level of importance to that, especially since the science and research is happening so rapidly. This is a good question from Ellen. Would a KRAS drug work on multiple types of cancer as long as a person had the KRAS mutation?
Yeah, so that's a basket trial theory and hypothesis, and definitely it'll be it's a theory that still is being tested through clinical trials. We have found that the answer is yes and no, because yes, the sotorasib, adagrasib, they're already proven to be drugs that can be targeting multiple types of cancers, right? So yes, you can get activity, but the activity levels seem to be different across the disease lineages, and the combination therapy also seems to be different for each of these diseases. So I think it's a partial yes. And then for each disease, you still have to tailor it to the specific disease. You may still use the same, one of the drugs may still be the same, but the combination approach may be different disease to disease.
Okay, I'm just going down these questions here. What do you think about mutation G12S and the NRAS gene in the cecum gut? I'm not quite sure I understand. Would you?
Yeah, so I mean, it'd be good to do the liquid biopsy to look at plasma circulating tumor DNA. It's just a blood test to look at this NRAS mutation in the blood. Whether this is specific to cecum or whether this is more clonal, meaning like most cancer cells harbor that mutation. So sometimes they can be subclonal, so only part of cancers have it, and maybe in the cecum, but not elsewhere. So it'd be good to figure that out. I think a liquid biopsy will provide the first clue, and then you get the allele fraction information and to see what's the dominant driver. If it turns out to be that specific NRAS mutation, you know, at the moment, we don't have a specific NRAS inhibitor yet. There are quite a few clinical trials being developed looking at pan-RAS, looking at the RAF dimer inhibitors, and these are still worthwhile considering in clinical trials. But outside of the trials, we don't have any approved therapy for NRAS.
Okay. G12V. The HLA. There is a G12V trial that I know has to take the HLA into consideration. I know Fred Hutch was very heavily involved in that as well. HLA, I'm sure that, you know, MSK does testing out the gate as far as, you know, what the HLA status is. But a lot of rather you're just kind of like run the mill, just not MSK locations, shall we say, are not necessarily testing for HLA, but that is a legitimate option. Are you familiar with that trial?
Yeah, so I'm certainly familiar with the approach, but I can't comment on that specific trial. The TCR approaches are certainly also being pushed forward at MSK. One of our leading physician scientists, Chris Klebanov, my colleague, is developing a series of TCR-based therapy, and they require HLA typing, so it's all matched. That's done pretty routinely at MSK in terms of our gene sequencing, and you'll get HLA information. But it could be also, it's a requirement for many of these cell therapy options. And often you have to take the blood and genetically engineer the molecule and then infuse it back. So-called adoptive T cell transfer therapy. Yeah, I mean, I don't know actually any trial that has produced, that kind of clinical data, the level of clinical data that was presented at ASCO. I don't think we're there yet, but certainly looking promising. And we've got case reports and anecdotes in pancreatic cancer, that was published in the New England Journal a couple of years ago. So there are some anecdotes, and I think this trial, I think I'm looking forward to seeing some early data from these trials.
Still pipeline is what I'm hearing you say.
Yeah, still early days. Okay. Phase 1.
Okay. All right. This is a good question. I'm busy. I'm just reading it. I am busy getting patients on KRAS trials. We always have the question, what is the best trial for the patient when they're offered a MEK or clamping RAS on inhibiting KRAS? Also pan-RAS, a better trial, or also is the pan-RAS a better trial because it inhibits all the pathways, or a specific targeted like a G12D versus pan-RAS?
Yeah, I think all of the above. If each of these give you a year or two, you times it by 5, that's 10 years, right? So I think you need to try all of that. The practicalities are often, the clinical trial match is very detailed and sophisticated, and it's very hard to get an exact match for the patient. So often these trials are just not a match, whether it's exclusion criteria, prior lines of therapy, whether it's HLA typing or whether it's the tumor location in the brain versus elsewhere, prior treatment history. There's a lot of these things are just not an exact match. So if you find a match, I'd go for it. And I wouldn't pick, I wouldn't be able to bet with a crystal ball whether KRAS multi is better than G12D specific. I'd want to try both of them, for my patients. So I think, and of course, the eligibility criteria often get in the way. Often, patients just can't get on because of certain history, certain, safety, presumed safety issues like pneumonitis. Some trials don't take any history of pneumonitis because of the concerns on this toxicity. And if the patient has some mild pneumonitis in a remote history, they might be excluded from this trial. So a lot of those, some of them are legitimate safety concerns, because some pneumonitis can be deadly. You can kill the patient on the clinical trial with a contraindication. Others are, simply just too rigid and too cautious, maybe too exclusionary. I think we need to continue to make the trials more accessible to the patients. And I think this is, the question, person from the Midwest, right? And how do we get more trials into the Midwest, right? We have to do more trials there. We have to partner with cancer centers and then go into the rural regions in the Midwest, right? Through technology, through telemedicine, through the remote monitoring devices, like we did in the COVID era, when it was all, patients were not coming into the packed waiting room anymore, and we ship drugs to the patients. So I think there's a lot of regulatory hurdles, and I think we need to, have that continued voice of access to clinical trials as a must. And a lot of these, state-based telemedicine rules they're clamping down on it. In COVID, we had a lot of freedom to navigate across different states, doing telemedicine. Now it's all restricted to your own state. They don't want to lose business outside of that state. So, I think we need to continue to have conversations with regulators, with policymakers, with lawmakers to just keep pushing on the message of clinical trials. Maybe they should create an exemption for clinical trials for all patients to get access. And the cost of clinical trials, the drug is for free, but in the US, the trial, the treatment, this all is all at cost. And the insurance is really heterogeneous. It depends on the level of your insurance. The out-of-pocket costs can vary greatly. Some of them are completely covered by insurance and others are not. So this becomes, certainly way above my pay grade and way above my expertise. But we need to still have that voice and keep spreading the word on the importance of clinical trials and that every patient has a right and deserves access to clinical trials. And how are we going to figure it out? I think everyone will have to play a role. And some companies are now increasingly taking on the cost of clinical trials in terms of other aspects of care, not just the drug itself, and reimbursement to hotel travel, flights. We have vouchers given to patients so the patient from the Midwest, it should have access to clinical trials. And I would still try to make a second opinion online and just try to at least explore these possibilities. I think one way or another, we'll find some ways to help you increase the access. And I do have patients who travel from all over the country and all over the world to come to see me one way or another. So,
Well, yeah, but
and
it's
sometimes,
you.
so
Well,
sometimes
yeah,
you just have to
and I
partner.
appreciate that. I'm from the Midwest. Okay.
Yeah.
Okay. Your accent
Great.
doesn't sound Midwestern to me. My accent is. I'm from the Midwest. And so I kind of feel that pain. And probably the reason the question is being asked is because it's right here, right now. We're not talking 5 years or even 5 months. We're talking right here, right now. And there is Kansas. Okay. There is Chicago. Okay. I know I can hear my own accent. It's for us back. And there are several in like Minnesota, Indianapolis. Okay. And there are different ones that are available. And just kind of like off the cuff, easiest hack to find out what clinical trials are available at a specific location, go on their website. So you can go on the Memorial Sloan Kettering website, see what clinical trials are there. Okay. And if for whatever reason, like you don't qualify, you're looking for something specific because you have, I don't know, something really specific, and you will know what they do not have that's available. The clinicaltrials.gov is a massive website. KRAS Kickers does have a list that we're going to be updating in a couple of days with the latest and greatest KRAS trials that we have that you can download as a PDF because it's just more manageable that way than trying to do a major full-on search. And to that end, since I'm talking about the work, I just would like to mention that we did have conversations about specific to the vaccine trials that are ongoing when we had our event. Dr. Marone from Hopkins was with us for the Kickers event back in March, and that stuff's all up on the website or on the YouTube videos. You can hunt up that way. Now, we do have a couple more questions, but it is the top of the hour, and I want to be respectful of your time because you're so generous as far as sharing your time, your wisdom, your expertise with us. Do you have a couple more minutes, or do you need to dip?
I can spend another 5 minutes or so.
Okay.
Just have a concurrent meeting to join, but I can tell them I'm a bit late. But I've put in the chat box a article from MSK about TIL therapy. So lifileucel is one of those first-in-class TIL therapies, and I think there's going to be a whole host of new therapies in that category, the class of medicines. So take a read. It's still pretty broad in terms of a class of lung cancer therapy, but you can see from the article, there's definitely some real promise in terms of patients' aggressive tumors being shrunk and patients living better and surviving longer. It's still early days. There's much more work to be done, and certainly we need to combine that with more specific KRAS inhibitors, and that's still, in the pipeline.
Okay. And then just kind of closing it all down. Do you have, I know all the rules and whatever, I don't know the rules. Are you able to do like second opinions through Sloan? Like, can we set that up online to have a meeting with you or what have you? Could you pop that into the chat box so we can share that with you?
Right. So yeah, so we have, it's all bogged down to these state regulations. So I'm only seeing patients in New York. So generally, back in the pandemic, anyone can get a consult through telemedicine, and we just do it. Now they're clamping down on telemedicine, so a lot of the consults will have to be on site or remotely in New York State. And then we have some written opinion options through like MSK Direct, through various channels, but then they get expedited access through sort of employer relations with MSK. So it's really miscellaneous. I see a variety of patients. I give opinion for a variety of patients online through that program. But I think it's best to just contact the MSK website. There's a new patient information. There's a hotline. You can just Google my name, MSK, you'll find it. And then, and definitely there are ways to help. And we have a long list of, we've got 1,400 doctors at MSK dedicated to fighting cancer. So certainly there's a subspecialist for every indication, right? And your indication your disease may not be the perfect match with my specialty, but certainly MSK can do a lot to help to find you the right specialist. But definitely if you have a local oncologist, we should also start there, because a collaborative approach often works better. I have a lot of patients from, including the Midwest, who come to see me, and they travel a long way, but I pick up the phone, I talk to their local oncologist. We come up with a plan that works for the local oncologist to give the patient the same treatment locally close to their home. Through collaboration, that's often possible, and also around the world. I often do that.
That's a fantastic way to kind of like end the series or end today's conference. As far as like second opinions matter, there's a lot of times that collaboration, it's not necessarily you're changing physicians, or you can collaborate. And so you're getting that level of touch from a high, amazing place like Sloan, in still living at your own home. Because honestly, at the end of the day, we're talking about living and surviving. So let me thank you immensely for being here and taking your time with us today and sharing your wealth of knowledge and like just your absolute heart, because I know how you feel about these clinical trials and how important it is, because we can do better, and we need to do better. And the only way that we can is by working together and making a difference. So thank you so much, Bob, for being here today and sharing with us. And I'm looking forward to look at all those hearts, all that love for you. We really do appreciate it. And I hope you're wearing the proper footwear today, because I know I haven't got you converted to stickers yet, but I know that you do wear socks.
Oh, so, yes, I wore that at ASCO very proudly. And thank you, Terri, for your leadership and your collaboration. It's truly an honor to be here. And I want to thank all the KRAS Kickers out there for keeping up your fighting spirit. You bring hope, not only for us, but for so many people around the world. Thank you.
Okay, thank you. Thanks so much. All right, everybody, thank you so much for joining us, and we'll see you on Facebook until we'll be on site in Hopkins in Baltimore on the 26th. So, and maybe next time I'll be able to come up to Sloan and do one on site. All right, y'all, take care. Thank you. Bye.
Thank you.